Hello and welcome to the third lecture in this series. This one is about molecular recognition in smell. But first, as promised, here's perfume of the day. It's Jiki by Guerlain, which is probably the oldest perfume composition that you can still buy. It dates back to 1889 and it took advantage of the then new synthesis of vanillin. It contains vanillin, lavender, citrus, and many other things that Guerlain has the secret of. And it smells wonderful. I use it as a sort of my own aromatherapy when I want to feel relaxed and peaceful. It is a fantastic fragrance. So today's topic is molecular recognition. So what is molecular recognition? Molecular recognition is the ability of a given compound, typically it's called a ligand, something that binds to something else, to attach itself to a protein, typically a receptor, but it can also be an enzyme, in a way which is specific and tight. The degree of specificity and tightness of the binding can vary enormously between one protein and another, or one ligand and another. But basically it's determined by the number of hydrogen bonds and van der Waals interactions that are formed between the ligand and the protein. The spectrum, the range of these interactions and the tightness and specificity of them is very large. But I'm going to take three examples that span that range so you understand what the problems that are specific to olfaction are. The first example, which is at one extreme of tightness and specificity of binding, is the binding of biotin, which is a small molecule, to avidin, which is a protein found in eggs, as a matter of fact. So what's special about biotin and avidin is that when you stick them together, they essentially never come apart. And if you look at why this happens, it's because biotin is able to make 7 hydrogen bonds with the protein avidin. Biotin is small, avidin is quite big, and when they bind to each other, biotin is connected to avidin by 7 hydrogen bonds. So that's roughly 20-something times thermal energy. And that means that once biotin is attached to avidin, essentially it'll never come off. And we know how exactly it works because we have the three-dimensional structure of avidin, the protein, attached to biotin. And we know that because with X-ray crystallography and with other methods now, you can get exact atomic positions for each atom in a protein, and you can see how one is attached to the other. And I can show you in green the structure of avidin, in red biotin attached to it. It's not so easy in this kind of three-dimensional representation to see how exactly the biotin is bound, but you can use a different representation, which is to flatten all the interactions into a two-dimensional plane so you can read them on a page and see what the molecule is connected to. And this is done by a website, a software called PoseView, which I find very useful. And the PoseView picture of biotin and avidin stuck together shows you very clearly the 7 hydrogen bonds, and that helps explain why avidin will retain the biotin essentially permanently. Now imagine that you're trying to do this with a receptor. In other words, you want to send a small molecule to a receptor and have the receptor be turned on by the small molecule. If it were like biotin and avidin, you could basically only do it once in a lifetime. You send the avidin and it's stuck there forever, so if the receptor is turned on, it's turned on permanently. And imagine that were the case for smell. You could smell one thing, and then you'll never smell that one thing again because the receptor will stay on all the time, and maybe you smell the same smell essentially forever. This would be completely useless. So you do want things to be rather specific and the stickiness to be good, so that there's a good affinity, meaning that a very small amount of the molecule is required to be around in order to bind to the receptors. But you don't want it to be too tight, otherwise it will never fall off, and the receptors cannot be turned off. So how is this done in practice? Well, in practice, with most hormones and neurotransmitters and other molecules that are found in the body that have to bind to receptors, what happens is you make fewer hydrogen bonds. 7 is clearly too many, so you try and make 4 or 5, and that guarantees, because hydrogen bonds are oriented in space, and they only work when there's a decent alignment, that guarantees specificity of binding, so that the receptor is not going to be turned on by any old thing that comes along. And at the same time, it guarantees that the rate at which the molecule falls off the receptor is still reasonably good, so that if you remove the substance from solution or from whatever external medium it's in, eventually the receptor empties itself of the neurotransmitter or the hormone, and you can start again. You can turn it on again with a second pulse. This is very important because what's called the bandwidth, in other words, the frequency with which you can turn off and on a receptor, depends on how fast the drug comes off. The drug or the hormone or the neurotransmitter. So as an example of this type of interaction, let's look at noradrenaline and its corresponding receptor. And you can see in this particular case that the noradrenaline molecule is bound to the receptor with 5 hydrogen bonds, which guarantees a good affinity and a decent specificity, but also make sure that the noradrenaline falls off when it's removed so you can start again. And that is useful not just for hormones like this one, but also in the brain where noradrenaline is a neurotransmitter, because if you have a neuron squirting noradrenaline onto another neuron, which they do, it's very important that the squirt gives a short response and not an extended response, because in most cases you want to be able to send pulses and have the other cell respond also with pulses. And if the affinity is too high, this won't work. The response will continue. Okay, so this is how it's done, roughly speaking, for the majority of hormone and neurotransmitter receptors. So let's talk about molecular recognition in the case of smell. What do we know about it? Well, from everyday life, we know that we can tell things apart by smell quite easily. You know when something is cooked, you know, you can tell one wine from another, you can tell one flower from another, you can tell when something is burning, etc. So we have a very good sense of smell that tells us, that can tell things apart from each other. And scientifically speaking, you can ask the question, how many different smells are there? And the answer is rather surprising. So far as we know, there's an infinity of smells. How do we know this? Well, we know it because if you ask a fragrance chemist, have you ever, in your entire professional life, encountered 2 different structures, molecule structures, that smell identical? They will say no. And chemists have been making smells since the 1860s on purpose, and long before that by accident. And to my knowledge, no one has ever said, oh look, here are these 2 molecules, different structure, identical smell. So the sense of smell has effectively infinite resolution. This is very hard to reconcile with the idea that smells will form rather non-specific interactions with receptors. Because how do you get this extraordinary precision in telling things apart from rather weak and non-specific interactions, such as, let's say, 1 hydrogen bond or 1 hydrogen bond plus some van der Waals forces? This is the fundamental problem of molecular recognition in smell. And it's a very peculiar problem because, unlike, say, with neurotransmitters or hormones or the molecules that I've shown you earlier, we don't have a single receptor for smell. We have 400. Mice have 1,000. So trying to understand how these 400 receptors each interact with odorants is extremely difficult. But there are a few advantages to this, which is that in a way, when we look at smell, we look at the average response of a large number of odorants. And the average response of all the different smell odorants to different molecules tells us something about what it is we're smelling. And perhaps the most important fact in all of smell research is that we can smell functional groups. What is a functional group? A functional group is an assemblage of a small number of atoms which determines the chemistry, the chemical behavior of a molecule. So for example, if I have a chain of a few carbons, let's say 6 carbons, and I attach on the end of it an OH, that's an alcohol group, that's the functional group of the molecule is OH. That means it will undergo certain chemical reactions typical of OH groups, regardless of what it's attached to. Similarly, I could attach an SH, a CN, an NC, an NOH, an NO2 nitro group, and a variety of other functional groups. Now, chemists are very familiar with functional groups because the functional groups really determine what they can do with the molecule. But they've also noticed that every time you put a functional group, whichever it is, on a molecule, that you get, the molecule tends to smell like the functional group, or more exactly, like the sum of the functional group and whatever the rest of the molecule would smell like if you didn't have it. So to give you an example, H2S smells famously of rotten eggs, as we've seen, but pinane thiol, so the pinane molecule, which by itself smells of pine needles, with an SH attached, smells of pine needles and rotten eggs at the same time. And that's a molecule contained in grapefruit. So we've got used to this fact that we can smell functional groups, and we find it sort of natural. And in fact, chemists describe molecules as nitrilic, sulfuraceous, smell like oximes, they smell isonitrilic, they smell ethereal. These are all functional groups. But if you think about it, it's totally mysterious. How is it that we can tell a functional group is in there? Well, a physicist friend of mine jokingly said, well, look, it's easy. When a molecule has sulfur, you can tell because the sulfur is yellow. But of course, the sulfur is not yellow. And what we're smelling is the envelope of electrons around the molecule plus the properties of bonds that these various atoms are capable of making. But think about it. If you can only make one hydrogen bond, how do you know that the hydrogen bond is being made by an OH rather than a NH, rather than a SH, or indeed an oxime? You don't know. It's impossible to tell. How can you tell? How does the receptor know? This is the fundamental problem of molecular recognition in olfaction. And the first person to recognize this fundamental problem was a chemist at DuPont Company, a very famous chemistry company in the United States, responsible for, among other things, nylon and all the other textile polymers, Kevlar, Mylar, etc., and for making the first synthetic musk. Now, Heinklopping's line of work was molecules that prevent the growth of mold on fruit. So he discovered thiobendazoles, which are used even today. They're sprayed onto lemons and various other fruit to prevent them from going moldy. But they're also useful because they kill worms in your intestines. If you swallow thiobendazoles, all the sort of parasitic worms tend to drop dead and come out. So, like many chemists, Heinklopping was interested in smells, I suppose, because he'd made quite a few in his professional life, like all chemists. And he was the first person to really point out this weird business that functional groups have different smells, whatever their molecular context. So he published this paper, The Odor of Small Molecules, in 1971, which was not particularly well known or much cited by other scientists, but which contains a table that describes the odor of some functional groups. And I find this table rather funny because he very accurately describes some of these smells. And in particular, there's one which after reading his paper, I was curious to smell myself, which is the isonitriles, which he describes as extraordinarily vile. And I can attest to the fact that they do smell absolutely awful. They're some of the worst smells I've ever smelled. They smell like I don't know how really to describe it, but sort of a metallic vomit smell would be a good approximation. And they're very intense. And there's just simply no way that anybody would ever find that smell pleasant. So here we are, we can tell functional groups, and we don't have a real understanding of how you do that, while at the same time having relatively few interactions of the molecule with the receptor, and relatively non-specific. This is the problem of molecular recognition in olfaction. So to finish off with an example, let's look at two molecules that are closely related in structure but smell completely different. One of them is cis-3-hexenol, which is an alcohol, as its name indicates, has an OH group, and smells essentially of cut grass. And the other one would be cis-3-hexene-thiol, in other words, 6,3-hexene-SH instead of OH. And it smells predictably also sort of like cut grass, but with a very powerful note of so-called sulfuraceous, in other words, the usual rotten eggs. Now look at those two structures. On the left you have the alcohol, on the right you have the thiol. They're fairly similar in size and shape, and they smell completely different. But here's the point. They smell completely different at all concentrations. In other words, there is no concentration at which the alcohol will have a rotten eggs smell, and vice versa, there is no concentration at which the SH will smell like cut grass. This is extremely strange and doesn't correspond to what we know about the rest of pharmacology. In pharmacology, that is to say the science of drugs, either man-made or natural, it is well known that when you replace an oxygen with a sulfur, as often happens in many drugs, just to try and achieve a slightly different affinity or potency or effect, you find that pretty much replacement of sulfur with oxygen or vice versa gives you an effect which is fairly similar, not identical, but no major difference. For example, barbiturates are very good sleeping pills and hypnotics and anesthetics. You can replace one of the oxygens on the barbiturate molecule with a sulfur, and you get thiobarbital, for example, which is also a very good anesthetic, though it's not chemically all that stable. So this peculiarity that O and S are totally distinguishable in molecules is unique to olfaction and is illustrated by this pair of molecules. Cis-3-hexenol, cis-3-hexene-thiol. So to make this clear, I can remove the color from both molecules, and you can see that in gray they start to look pretty similar. Well, you might ask, well, fine, that's the shape of the molecule, but what about the molecular dynamics? Do they move differently? Do they do something different? Well, not really. The molecular dynamics shows that, yes, they jiggle around in much the same way. The configurations adopted by the molecule are rather similar. There's nothing obvious that would enable a system to detect the presence of a sulfur as opposed to an oxygen with complete precision and infallibility at any concentration. And that's the problem that we will deal with in the next few lectures.